Toxoplasma gondii is a single-celled parasite that causes a disease called toxoplasmosis. Infected cats can give the disease to humans when they shed cysts containing the parasite.
Toxoplasma infects many people worldwide. The estimates range from 10% in certain populations to upwards of 70% of individuals in others.
While the infection stays dormant in most people, it can spread and become life-threatening in people with weakened immune systems.
Toxoplasma is quite good at getting into places it shouldn't be, like your eyes or your brain. And once it's there, it focuses on eating and replicating as much as it can, in the process destroying those sort of very important organs.
But Toxoplasma is also able to change into a dormant state and resist the immune system for years.
So there are all of these open questions around how the parasite is able to evade the immune system, how the parasite is able to survive for such a long period of time undetected, and then how it is eventually able to reactivate from that state and cause disease in people who become immune compromised.
One key question is exactly how Toxoplasma controls its transformation from its fast-growing state to its resistant, slow-growing form. This process is called differentiation. The form of Toxoplasma that first invades human cells is called a tachyzoite.
So tachyzoites are replicating very quickly. They're infecting host cells, replicating inside that host cell for a couple days, and then exploding that host cell and finding new ones to invade.
But once inside a cell, the parasite can then differentiate into a second form called a bradyzoite.
And what's tricky about bradyzoites is they actually insist, so they form a very resistant wall around themselves inside of your cells, and they stop growing as quickly, which means that they don't actually get targeted by many of the therapeutics we would normally use.
Scientists have been trying to figure out what molecular mechanisms Toxoplasma uses to control differentiation. For some time, it seemed that a number of proteins called transcription factors allowed the parasite to activate genes needed to make the transformation.
The field had arrived at this consensus that because of the complexity of that differentiation, it was likely dependent on several transcription factors working in unison, all contributing minor effects to the overall transition.
But Laredo and Waldman suspected that there had to be a single main control switch for this process, called a master regulator.
If you think about differentiation, it's this massive change in life cycles. You want to decide to differentiate and you want to commit. And to do that, you need to have a tighter set of transcriptional control for that process. It's kind of hard to do if you have that many players.
The researchers developed a strain of Toxoplasma that would signal when it was differentiating by glowing green.
What I did for a lot of my early years of my PhD was trying to get a reporter strain established in Toxoplasma, something which would sort of read out whether these parasites were trying to differentiate or not.
Then they used the gene editing platform CRISPR-Cas9 to knock out a set of about 200 genes that could act as transcription factors. They looked to see if any gene seemed necessary for the parasite to differentiate. And there was only one gene that, when lost, completely prevented the parasite from differentiating into bradyzoites.
Almost nothing in these libraries had any effect on differentiation at all, except for this one gene that we found. We've named it BFD1 for bradyzoite formation deficient 1.
We could show that it was not only important that when breaking it, we broke that circuit, we broke that transition from the acute state to the chronic state, but also that upon upregulating it, we could artificially induce that switch. And in my mind, this is sort of where biology meets engineering. Our ability to actually control that transition through the synthetic expression of that particular factor really shows us that we've gotten it. We've gotten the key bridge between those two different states of the parasite.
The master regulator of differentiation offers a new target for treating chronic toxoplasmosis infections.
Finding what we call the master regulator of this process was both surprising and exciting, and I think it'll help us really understand in a clinical scenario how we can interfere with that process.
The findings highlight the importance of basic biology research in finding new ways of attacking problems to treat disease.
I think basic biology is incredibly important to advance human health long term. To be able to combat something in a rational way, you really need to understand how what it is you're fighting works. if you're trying to understand how to stop a process or how to reverse something like chronic infection, you need to understand how they got there in the first place. And so by doing this sort of basic biological research, we can identify what those targets are, and by understanding the mechanism, you can really understand how to try to interfere with or reverse that mechanism.